I'm going to record it right now. Thank you. All right, so this is chapter 3, and we are looking at the cell. All right, what we wanted to do, we wanted to travel inside of the cell. All right, we wanted to find out the important department within the cell. All right, so and we will learn about some of the concepts and function of these as well. All right, so let's get to it. Um, a cell consists of the cell membrane, all right, and within the cell membrane, just 1 second. All right, so within the cell, there is a cytoplasm. When we say cytoplasm, basically all these areas within the cell is called the cytoplasm. Now, sometimes you hear something known as a cytosol. Cytosol is basically a semi-transparent fluid in the cytoplasmic material that basically is the mixture of colloid and other solution. Basically, what it means is the fluid part. This could be like, water along with other stuff that is in the water, sugar, salt, things in that nature. So the content of it is called the cytosol. All right. Um, you have what's known as uh, organelles. Organelles are different departments. So, for instance, you have a nucleus. In anatomy, you learn you have like a mitochondria, for instance. So these are called organelles. Organelles, they have, it's kind of like a department. If you go and kind of like think of the cell as a complicated biopharmaceutical company, the biopharmaceutical company has a different department. It has shipping, receiving department, marketing department. It has many different departments, all right? Accounting department, research and development department. So those are kind of like an organelle. There are so many different organelles within the cell, and each of these departments, they have their own job. They have their own, they do their own task. And together, all these departments are important for the cell, all right? So let's go ahead and take a look at the mitochondria. Mitochondria is a sausage-shaped structure. And what it does, it generates energy for the cell. This energy is also known as a powerhouse of the cell. All right. So the energy that is produced is called ATP, adenosine triphosphate, we discussed. And the cell sometimes need the energy in order to transport substances in or out of the cell. Sometimes the ATP is needed to allow the cell to make protein, for example, that last time we discussed using the DNA. All right. So a lot of things requires the energy, all right, in form of ATP. Now, interestingly enough, this is also known as a site of cellular respiration. Means the cell is actually breathing. The reason is basically they take out oxygen that we get from our respiratory system, and they give off carbon dioxide. Carbon dioxide that we breathe out is all ultimately coming from the mitochondria. In the process, you need sugar or glucose as well. All right, so we need the glucose, and the glucose, due to a lot of biochemical pathway, which we're not going to get into at this point in time, they're very complex, and when you take biochemistry, you will go towards it. But basically, what happened is there is a lot of uh, aerobic respiration. Aerobic respiration means they need the oxygen in order to do this. And as a result of that, they generate energy, usable energy for the cell. All right. And they also can make water, energy plus water. All right. Now, if we go ahead and take a look at the mitochondria itself, all right, mitochondria itself, they have a circular DNA, they have ribosomes. Ribosomes are involved in protein synthesis, and mitochondria can give rise to more mitochondria. So, in other words, they can give rise to more mitochondria. All right. So a lot of times scientists believe that at one point in time, these mitochondria was just kind of like a bacteria, a small microorganism. They got into our cell, find shelter, and they were able to produce something that we really need. All right. The fact that they have their own DNA and they're capable of dividing and so on suggests that. All right. Interestingly enough, this DNA comes in only maternally. All right. So we get our mitochondria from the mother's side. The story goes is this. Here is the egg. The egg consists of genetic material or nucleus, which contains the chromosomes and so on. And here is the sperm. The sperm in anatomy, you probably remember that. All the mitochondria happen to be in the midpiece. It energy that produced for that helps to propel the tail or flagellum. And then this head part later on in time is the only part that penetrates all the mitochondria and from the father stays outside. So what does that do? Inside of the head region, there is a chromosome, half of the genes and the chromosomes from the head region combines with the genetic material from the female, and you get the full set. That's why you are a combination of half from you, half from the mother, half from the father. Huh? All right. So if you want to, you could text your mom after the lecture and say thank you for your mitochondria if you like. All right. Next up, we go to the ribosomes. I cannot write again. I don't know why this sometimes happens. All right. I don't know how come I cannot draw. This pen is not working right now. Okay. I'll continue just for the time being. Maybe I won't draw for now. Uh, the pen is not working, but that's okay. So let's go ahead and take a look at the ribosome. Ribosomes are a site of protein synthesis. All right. Now I can maybe. There you go. So ribosome is the location where all those amino acids put in together. Okay, there are free ribosomes synthesize soluble proteins. There are ribosomes that are attached to the other areas, such as endoplasmic reticulum, and the job of the ribosomes are to facilitate and is to make the protein this location. All right. Ribosomes are being manufactured inside of the nucleolus, which happen to be inside of the nucleus, and they are involved in that. There are two subunits, and inside of them lies our RNA, which we haven't talked about different types of RNA. We have three different types of RNA. You have messenger RNA, you have rRNA, and you have transfer RNA. I need to revisit the protein synthesis to give you guys the job of that. But what is important right now is that rRNA is part of the ribosomes. If you recall, we went from DNA to messenger RNA to the protein. So let's have a messenger RNA. So just for the sake of it, AUG, GGG, let's say CCC, all right, let's say, uh, let's say AAA, just making it simple, all right, and finally, let's say we have ACT, U, no, UGA. Sorry, this is RNA.
Professor, I have a question before you continue.
One UGA. All right, hold on for your questions. Let me finish this part and then we'll talk about it. So this is a messenger RNA. If you recall last time, we had the DNA and we learned how you can transcribe using the base pair rule that I gave you and to get the messenger RNA. I gave you guys activity to do in the class. My expectation is that you already know how to do that. Now, let's see. And we also mentioned every three of these is called triplets and is a coding for a particular amino acid. Uh, ribosomes. Ribosomes, there are two subunits. One subunit sit in here because they have a rRNA, they kind of like match in there. All right. Another subunit goes on top. All right. And these subunits, they have two regions. All right. They have one region right over here, and they have another region right over here. And these regions are designed for fitting what's known as a transfer RNA. Now, transfer RNA, again, is RNA. Transfer RNA or tRNA, they possess what's known as anticodon. Anti means opposite, codon. So it's code for the opposite of the codon. So if you remember AUG and using the universal coding system, if you look at those chart, it's supposed to be code for methionine or met, all right? But in reality, there are many different transfer RNA, and one of these transfer RNA that brings in the met, all right, have an anticodon of UAC. So the UAC, this whole thing is going to fit in here. So let's bring it down here, and it's going to fit right here, UAC. See, they formulate hydrogen bonds with one another. U pairs with A pairs with U, C pairs with G, G pairs with C. But in reality, these are the anticodon. This is the anticodon right here is the anticodon. All right, and this is the transfer RNA, and transfer RNA bring in the methionine. All right. Yes, there are a lot of enzymes and other things that working, but let's go through the next one. The next one, uh, have an anticodon of, if you want, you could pause the video later and figure out if you remember, have an anticodon of CCC, and therefore you have another, it brings in another amino acid, in this case, glycine, gly. So here's what happens. Watch, right here, bond forms, especially if you have the proper enzyme and everything, new bond form. This bond was a peptide bond. Peptide bonds were covalent bonds, basically. And what happens to this other bonds? It breaks right here. And what happens to this transfer RNA? Well, this transfer RNA is going to depart, all right, and bring in another uh, amino acids later on. But now you just attach the next amino acid, which was glycine. All right. Now this ribosomes is going to shift. It's kind of like a zipper that you're zipping thing through, is going to go to here now. So I'm just going to redraw it in here. And this is the ribosome on the upper part. And I'm going to try to erase this part, you see. All right, this one got departed already. All right, so we no longer have this. Now the next transfer RNA comes in. So at the beginning where everything gets assembled, this is sometimes it's called initiation. The ribosome subunit comes in attached, all right, because they have rRNA in it, and they can find out where the AUG is and so on, and they attach. Transfer RNA comes in with their amino acids, and this is called initiation. The second step uh, is elongation. That's what we're doing right now. See, we are putting these amino acids together and we're going forward. What are you doing? You're elongating the chain of amino acids, which are here. All right. Uh, I'm going to see if you guys can maybe give you a different transfer RNA now. These are 3 transfer RNA, and they have different anticodon. I'm just going to call this A, B, and C, just for arbitrarily, all right? And I'm going to put in the following. So you guys tell me which one fits next, all right? Okay, A, B, C, D, or E. Which one of these fits in here? What do you think?
B.
E.
B?
E.
Okay.
B. B.
Okay.
Yeah,
So basically,
B.
this one, it has to have an anticodon because this is a codon, all right? And G, 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 and basically this one, I think, is bringing proline, all right? And then again, next bond, peptide bond forms. This bond is going to break. This one departs and gone. And you can see how we are elongating, all right? Next, the ribosomes is going to shift, and we are going to the next one, all right? And here is the next one. What do you think? Which of the following transfer RNA, you can select none of the above, is going to fit in here? What do you think?
E.
E.
B
E.
as an Edward, right?
Yes.
Okay, you're right, because this one is triple U. All right, make sure you understand this part, okay? So the triple U comes in, and basically is attaching the next amino acid, whatever it is, they're bringing it. They have the anticodon. The anticodon meets with the codon, and again, the new bonds is forming. And finally, you get to the stop codon, which is UGA. And at this point in time, there is no more elongation. In other words, everything disassemble, and that is called termination. All right. Uh, again, I mentioned this to you a lot, but yeah, a lot of these things you guys can seek up in YouTube videos, and let's see what we can find. And it would be a good idea for you guys to do further studying. Don't limit your knowledge just based on what I tell you, because visualizing it, seeing it, and all that might make a big difference. I just going to maybe search translation.
Professor, there's a series that called Crash Course, and I shared the link yesterday, and they have a really good video on translation. I don't know what your opinion on that is.
I have checked it out already. It's pretty good. They have a lot of the other crash courses and so on. They're pretty good. I know exactly which one you're talking about. So they're pretty good, and I thank you for sharing that. Great job. But remember, the crash course, they really go so fast and everything. I would say first read the material, then look at the videos, look at the notes, and listen to the lecture several times, and then look at the crash course. I think it's going to make a lot more sense than just like looking at it. That's just my opinion, but you're welcome to do whatever you like. Okay.
Okay, sounds good. Thank you.
Yeah. So here is the YouTube video.
I shared this one yesterday.
You share which one?
This one, the one you're playing.
Okay, good job. Thank you. So you guys, this is another one you could take a look at. I thank you. I just looked it up, so I didn't know which one, the one that you shared. I didn't get time, but there are many of them, and you guys can go ahead and take a look at it. This one is 7 minutes long, so it tells you like the one that has a lot of views are usually good ones. Like this one is 7.5 million people looked at it. It's from 5 years ago, and it's only 2 minutes long. So since you share that one, we can look at this one.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free base.
The polymerase ends with ASE. So this is kind of like an enzyme we discussed. It just facilitates this, okay? And I didn't go to the details, but I think you should know that as well. It's really easy. Basically, it's just match pairing them.
In the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA.
And notice that, again, same thing as we discussed earlier. This is the coding strand right here, and these are the new messenger RNA that are being assembled one by one. So U pairs with A, you can see that, all right? So it's, and we discussed about the base pairing.
This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid.
This is a transfer RNA, and these are the anticodon, folks. This is the amino acid that is bringing. Now, you could see this is probably a better diagram that I draw for you, but it's a similar concept.
Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid.
Notice that these circular structure are amino acids. So they're bringing amino acid and new bonds are being made. What kind of a process do you think this one is? Do you think this one is initiation, elongation, or termination? Elongation. Good job. Good job. So that's what it is. At the beginning, when everything assembles, that would be the initiation. When everything disassembled, that would be the termination. So pay attention on it. Take a look.
This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
All right, so that was a good example, and you know it goes on, and there's so many useful videos you could go ahead and take a look, and you know it just depends on how much time you have. But I strongly encourage to understand the material, so when you go back to the lectures and so on, it will make more sense to you. So all of that happening in the ribosomes, folks. All right, that's why we call it the site of protein synthesis, because all those activities happens, the elongation, adding all these new covalent bonds or the peptide bond happening over there. All right, moving, going forward. Now, let me go ahead and pause the video and see if you guys have pause the recording. And let's go ahead and take a look at what happens. We have other organelles to take a look at. So here you have the cell. Here you have the nucleus. Continuous with the nucleus, there is a set of maze-like structure. They're called RER. But a lot of those tiny ribosomes we just talked about, they are located, these are the ribosomes, right on it. So these are called RER, or rough endoplasmic reticulum. And the rough endoplasmic reticulum, it just have an appearance that they are rough, that's all, okay? But technically, they have several function. Main function, obviously, is these type of cells that they have a lot of rough ER, they must make a lot of protein, all right? So the cell membrane factory, where the integral proteins and phospholipids that form part of all the cellular membrane are being manufactured, all right? So the enzymes that catalyze the lipid synthesis, you know, all of those happen to be related into the rough ER, rough endoplasmic reticulum, all right? So over here, you're making the protein. This protein later on could be the enzyme, for example, or other things that we discuss. Now, another structure nearby, which I'm going to show you in a different color, and you could see this maybe a better drawing, maybe a little bit later, all right? So these, they don't have the ribosomes on it. So these are known as a SER, all right? A smooth endoplasmic reticulum is the name of it. And the reason is they don't have the ribosomes on it. So they don't play any role in protein synthesis, but the smooth ER can actually have many other functions. One of the function of the SER happen to be they can synthesize cholesterol, synthesize the lipid components of the lipoprotein in liver cells, for example. They can metabolize lipids, for example. So I just put in maybe synthesize cholesterol and phospholipids. A smooth ER, they can play a role in absorb, synthesize, and transport of fat in a small intestine. They have many different jobs. They have detoxification job, detoxification of the drugs, for example, or certain pesticides or chemicals in the liver and kidneys are their job. By the way, a lot of medicines that people take, they're not really without any side effects. There's a lot of toxicity inside. In fact, if you go and take a look and read the Tylenol bottle, it tells you it could cause liver failure if you take in a high dosage. Consult your doctor if you need to take more dosage. What does that tell you? That means, it's a toxic material and your liver is going to pay the price for it. So be careful when you're taking medication, especially if there is no reason for it. Anyways, they can break down or store glycogen to form free glucose in liver as well. Another job of the SER, especially in the muscle, they can store calcium. All right, and very soon you'll find out as well. All right, because we're going to go to the muscles. All right, so here you got our cell, and over here we got the nucleus. All right, and there is a part right over here that looks like a bunch of pita bread on top of one another. These are called Golgi. All right, or Golgi apparatus or Golgi body. All right, and what happened is that those proteins. Remember, we discussed that earlier. We discussed that the rough ER, for example, making the proteins and so on. Well, some of those proteins, they are placed into the vesicles. And these vesicles basically is like a bubble shape containing those amino acids. They can come towards the Golgi, all right? So they have a receiving end, and then they have a shipping end. They can get processed and they could be shipped outside, or they could get processed and go into another destination, all right? So basically, Golgi body, I would say, is kind of like a shipping and receiving department of the cell, because it has receiving end, it has shipping end, and it packages the protein, modifies the protein, and send it out to the proper destination. All right, that's the job of the Golgi body. Next up, let me see what time is it. You guys, it's about, let me see. Concentrate on the other organelles. We're almost done. So the next organelles I would like to take a look at is lysosome. All right. Let me go to the new page. A cell sometimes there's like a circular structure with digestive enzyme inside, and this is called lysosome. Lysosome has many different functions as well. So, but the enzyme can digest almost all kinds of biological molecules. So, this usually contains what's known as an acid hydrolysis, and the lysosome function is basically demolition crew. Think of as they break things down. You could break down food, you could break down bacteria, viruses, even some of the organelles that we discussed. Say, for example, here is the mitochondria. They could uh, fuse with the lysosome and they could be broken apart. So it can have many different functions. Because of this enzyme inside, whatever goes in there, it just breaks it down. And sometimes this enzyme can actually self-destroy the whole cell, all right, because it's going to come out and pretty much degrade uh, and uh, self-destroy by degrading and breaking down some of the membrane. The cell no longer can function, all right. So it can have a lot of different things breaking down and such, and lysosome is usually abundant in our immune cells, as you can see why. We also have it in digestive cells, as you can see why, all right. And so they can protect us. If for whatever reason the lysosome is not working properly, then you'll have a massive infection. The infection can go around and just multiplication and causing a lot of problems. All right. Interestingly enough, there is another organelle. Is called peroxisome. Peroxisome are a spherical membrane sac-like structure, and they also have variety of powerful enzymes, which is mostly oxidases and catalases and such. So to just giving you an idea, they do a couple of things, but the major one is they also do detoxification, all right? Detoxify or detox, all right? Harmful substances, detoxify, harmful substances, all right? A lot of these harmful substances could be like alcohol, formaldehyde, but most important one that they do, they neutralize free radical. Free radicals are lone pair of electrons. They're highly reactive. And what they do is these unpaired electrons on the outer shell, what they do is they can scramble, they can mess up other biological molecules. So oxidases, they just convert free radicals to hydrogen peroxide. All you really need to do is think about free radical as unpaired electrons that are really reactive. So when they are really reactive, they could react with other molecules such as DNA, for example, and they could modify it, and that causes genetic changes, for instance. They can also be useful in detoxifying harmful substances as well, especially in the liver and the kidneys. Peroxisomes are very numerous where they do the detoxification and all, right? Now, we went over several of these organelles. Interestingly enough, all these organelles, they're not just working by themselves, but they work as a team. All right. There are a lot of things that are happening. Like, for example, you need the nucleus. Nucleus contains the genetic material. Nucleolus can make the ribosomes. This genetic material from DNA, you make him RNA. RNA comes out using the ROF-ER and the ribosomes to make the protein. The protein then can go to the Golgi apparatus, get modified. They can be transported out, or some of these proteins like the enzyme that they contain, they could put it into a lysosome, they could put it into a peroxisome, and they could do their jobs and so on. And in order to do all those things, sometimes you need energy, so you need mitochondria. This is kind of like remind me of many different companies. If the company has a great product, but you don't have a good sales force, nobody even know you have a good product because your marketing is weak. On the other hand, if you have a superb marketing, but you don't have a good product, again, you could have affected too. So a lot of these things are related to one another, all right? So just like the school that has different departments, just like the workplace that has a different department, just like community that has a different department, all right? Uh, they're all there to work towards the common goal, and that is to ensure that the cell is doing what they're supposed to be doing, and all that. Next up, we have cytoskeletal. Cytoskeletal is kind of like a frame to a house. It's the skeletal of the cell. All right. Without the cytoskeletal, the cell can collapse. It could have a structural damage easily, and you're not having the support that you need. Also, a lot of these cytoskeletal might play important role in movement purposes, especially when we're talking about the microfilaments in the muscles. Those are also important, but it comes in three different shapes: microtubule, which is the largest; microfilament, which is the smallest; and intermediate filament, which is something in between. All right. You could go ahead and take a look at what they look like, both on the microscope and also kind of like figuratively. And starting off with microfilaments, this is a good example of it. You could find it in the muscle. And they kind of like showing in here too. This is the microfilament. As you can see, microfilament is the smallest one, all right, in terms of the diameter at least. Then you have intermediate filament. Intermediate filament, they do have a role of anchoring other organelles together so that the organelles are maintaining, and they're kind of like small ropes that are tough. They're insoluble protein fibers. And finally, you have microtubules that are largest one. Microtubules, they have many, many different roles, but they're like a network, and they provide a lot of structural support. If you go ahead and take a look at all of these, there are more details of them in your book, and also more detail of them on other resources that you look into. I'm just making it as simple as possible. Uh, uh, for example, when you are dividing the cell, you wanted to move the chromosomes in the different areas and so on, you need microtubule for it, all right? So microtubule is also determining the overall shape of the cell as they get distributed in the cellular organelle. So either way, if you look at the construction buildings that are building up near you, you'll see that there are a lot of rebars. Some of those rebars are really huge. Some of the rebars are smaller in diameter, and there are actually some kind of a number to indicate what kind of a rebars they have and so on if you want to purchase one. It's a similar concept. In construction, you wanted to have a sound structural support for everything. All right. Next up, we're going over the centrosome and centriole, maybe. Yeah, centrioles, these are a small barrel-shaped structure or organelle that all you really need to know is they are being used during the cell division. They're coming into two different forms, cilia and flagella, and again, also for movement purposes. Right over here, you can see what they look like. They are really involved in during the cell division, and they can manufacture some of these microtubules. And again, like a sperm tail, for example, you know, they're made out of microtubule. All right. So you could see how they could play a role in movement, for example, that we discussed. Right over here, this showing you a structure of the cilia. Certain cells, as you know, they have these cilia, like, you know, the cell in your trachea, for example, has cilia to trap the dust particle, to distribute the mucus lining. Your egg cell has cilia that therefore when you are ovulating, the cell can go from one region to another. So anyways, the idea is that they have a lot of these microtubules within them, all right, and that's what it is. All right. This particular case is showing you the movement of the mucus across the cell. So this mucus right over here, they are being distributed using these cilia. They're moving and so on. Surprisingly, you know, this cilia's function or the movement, it gets halted by people that are cigarette smokers. You know, when you are smoking cigarette or you are a smoker, these cilias are paralyzed. They don't move as well. All right. So you don't distribute the mucus. So a lot of times when substances due to breathing goes into your lungs, people that are cigarette smokers, for example, they start coughing a lot. Coughing because you cannot get them out anymore. If you had these functional cilia, you could kind of like propel them and move them out of your lungs. But if you can't do that, you got to constantly cough. And that's why they do that. All right, the rest, I kind of like went over it. So right over here, you could see the nucleus. Inside of the nucleus, you have the genetic material. They are organized right now with a lot of protein and so on. So you don't see them, but they're organized in the chromosome.
Oh, we can't hear you, Professor.
Okay, yeah. Yeah. I wasn't saying anything. Thank you for reminding. I was just pausing it for a moment. So those are the organelles that you are responsible for. Let's go ahead and take a look at the process of mitosis now. So in this idea is showing you how the DNAs are organized and into this fundamental unit of nucleosomes. It's just a way to concentrate all these DNA and collect them into a form of chromosomes, as the video was showing you as well. This is a cell cycle. You need to know about the cell cycle. The cell undergoes division. But before the division, a lot of things happen. All right. And I would like to go over the cell cycle really quickly. Many of you guys had this in anatomy, but because it is physiological roles that we need to cover, I think it's essential for you to understand this part as well. All right. So majority of the time, the cell happen to be in the interface. I know this pie chart is not really telling you the whole story, but 90%, approximately 90% of the time, the cell is in interphase. What does that mean? The cell is resting. The cell is preparing for division. The process of division has not occurred. These are all what it means during the interphase. So growth is part of the interphase, G1, and synthesis, the S phase. This is usually pertained to the DNA synthesis or chromosomes duplication. And finally, you have a G2 or the final growth, final preparation. So it's kind of like a washer and dryer. When you put in your clothes and you press the button, there are certain phases that every time it has to take place, like adding detergent, mixing it with water, rinsing, and spinning. Every time you turn on your washing machine, there are those specific steps, one after the other. So these are exactly what happens. First is the G1 phase. During G1 phase, there is a lot of growth. There is a lot of protein synthesis. There is a lot of metabolic activities. During the S phase, chromosomes duplicate. Now remember, we have 46 chromosomes. In order to put in 46 chromosomes in the daughter cells, you need to multiply this DNA. And with this, kind of like talked about this already, is Xerox copying a DNA. If you remember, one strand was kept as a template and the newer strand was being made. This is exactly what happens, but in a very large scale, because we have 46 chromosomes and each chromosomes contains so many different DNA bases. So you basically Xerox copy them. When? And during the S phase. So if we have a 46 chromosome during the S phase, now we have a 92, it doubles up. If a particular organism has a certain amount in this phase, S phase, it doubles. So notice later on, you'll see this, but notice that they're kind of like an X mark. Why? Chromosome does not look like an X mark. Chromosome is kind of like that. But because they Xerox copied, that's why they look like X mark, okay? But they're still attached to one another. You get to a point that they get separated, but they are attached to one another. All right, so the last step is the G2 phase, which is final preparation. At this point in time, the cell is ready. There's a lot of protein synthesis. There is a lot of enzymes. There's a duplication of a lot of different organelles already occurred, and they're about to enter mitotic phase, which is the actual division side. Approximately 10% of the time, the cell remains into the mitotic phase. What that means is, the cell takes a lot of time growing and preparing, but the division part is rather quick. Mitosis and cytokinesis is part of the mitotic phase. Cytokinesis is the division of the cytoplasm. So when the cytoplasm is dividing from one another and no longer are being shared, that's called cytokinesis. So notice that cytokinesis is about to happen. As soon as it happened, that phenomena is called the cytokinesis. What about the mitosis? Well, this is again review of your anatomy, but we discuss about prophase, metaphase, anaphase, and telophase during the anatomy, and I would like to go over it one more time. Again, I will encourage you guys to see some of the videos that are in YouTube. All right, just type it up, mitotic phase or mitosis, and it should be able to give you guys a lot of related videos. You watch a couple of them and you kind of like master it really fast. But we can go ahead and take a look. And by the way, when you do that, then when you go back and listen to the lecture, it will make more sense to you. All right. So this is just showing you how the DNA replication is occurring. I didn't cover a lot of enzymes and a lot of molecules that coming into a play. So because I wanted to make it as simple as possible. All right. But basically what happens, you separate the two strands from one another using certain enzymes. All right. And what happened is you're keeping the old strand as a template and you're making a new one. That's what happens. In order to add things at the basis, you need an enzyme known as DNA polymerase. All right. But there are different kinds of DNA polymerase as well, like DNA polymerase 1, 2, 3. Don't worry too much about that at this point in time because I'm not really focusing on molecular biology stuff. But what happened is, you keeping old strand, you're making a new one according to the base pair rule that we discussed. All right. A pairs with T, G pairs with C, and that's it. Pretty much it. All right. So you need a DNA template. All right. You need a different type of enzyme, polymerase. ACE ends with ASE. DNA polymerase. That means it's related to the DNA, making DNA. All right. So that's what you do. You're doubling up. All right. There are other things that you start off with, like RNA primer, and later on, ligase and other things that we're not going to get into right now. So why do we need to have mitosis? Should I just remember, okay, PMAT, P prophase, M metaphase, A anaphase, and T telophase, and good to go? Maybe for anatomy was good, but you really need to know why. Why are we learning about mitosis, and what is the job of mitosis? Mitosis is identical cell division. So you starting off with the cell that consists of 46 chromosomes, you're going to end up with cells that are 46 chromosomes. They are identical from one another. All right. So now that are identical, you could think about the job that they do. A lot of times in our body, cell get damages. Think about, like, maybe you were playing soccer and you fell and you had bruises on your knees. The cells around them, they need to make those cells. One cell become two cell become 4 cells. That might take some time, but pretty soon your injury is going to be healed. Most of the surface of the skin consists of epithelial cells. Epithelial cells, they have a high mitotic capability. So if you went to the, for example, beach and you got some sunburn or whatever, after about a week or 10 days, nobody even know that you had sunburn because, the skin, got replaced. All right. So cell replacement, cells that are damaged, cells that are old, cells that need to be replaced. That's one of the jobs, cell replacement. Another job that I can think of is growth and development. Growth and development, since you were born, or even while the embryonic development was taking place, they're all mitosis. The cells are multiplying. In fact, if you take any of your cells, your DNA in the cells of your saliva is the same as the DNA of the cells on your skin, same thing as your DNA is, in your nerve cell, etc. So they're all the same. Then why is it that they look different then, if they're genetically the same? Well, they look different because they're programmed different, okay? Different genes get expressed in different cells, all right? And there is a lot of detailed and complex activity that are taking place, okay? So the last one, it really doesn't pertain to our system, but asexual reproduction. Not all organisms, they require a sperm and egg in order to multiply. Some organisms, they can give rise to their own, and they are identical genetically to their mother or father or whatnot. There is no mother and father. We could just call it parental cell, all right? So what is happening? During prophase, I'm going to go over the steps now. During prophase, all right, this is the early prophase. The pair of centrioles, remember the centrioles were playing a role in cell division and they were making microtubules. So you could see already this mitotic spindle, these are microtubules, they're start forming and they're going in the opposite direction, all right? The chromosomes, you could see, now we could visualize it. We could see it. And this is a different microscope. If you haven't done your lab, one of them was about the microscope and different microscopy, electron microscopy, compound microscopy, and so on. So you could see the chromosomes in blue, all right? Now, we're still in the prophase. Towards the late of the prophase, this nucleus is going to degenerate, all right? So you could see the nucleus is already degenerating. These are the remnants of it. These are the fragments of it. And at this point in time, the spindle centrioles are completely opposite of one another. You could see right here, and you could see right here, and they're generating the microtubules that we discussed. And these microtubules, if you look carefully, if I could show it to you, maybe a larger scale, you could see they are attaching to the chromosome. This area is called kinetochore. Why are they attaching to the chromosomes? So that way you could move these chromosomes, all right, and line them up. And that's exactly what happens on the next phase. The next phase is called metaphase. Metaphase means middle, and the chromosomes are lining up exactly middle of the cell, all right? Next up is anaphase. Anaphase, the daughter chromosomes separate. They're identical to one another. Now they're going opposite direction. Cell is elongated. Notice that the cell is elongated. They're going in the opposite direction. Look how elongated it is. All right. And these microtubules are pulling these daughter chromosomes. That's called anaphase. Finally, we got the telophase. Usually the cleavage furrow is apparent. And the telophase is kind of like opposite of the prophase. In prophase, nuclear envelope disappeared. In here, they start reappearing. In prophase, the spindle fibers appeared. In here, you could see they are disappearing. So I should probably erase that. They are disappearing, all right? And the chromosomes, they're going to go back to their condensed form. So in other words, they're going to be incorporated with a lot of different proteins that they're attached to. And the cell is about to get pinched into two cells. All right. So cytokinesis, as long as the cytoplasm is still being shared right over here, cytokinesis has not been completed. The moment that phenomenon, as the cell kind of like divides and this part with this part, no more cytoplasm are being shared with one another. This is called cytokinesis, division of the cytoplasm. All right. So the rest, the role of the RNA and how does the protein is being made and all that, we already discussed. So that summarizes pretty much the important aspects of chapter 3. And there is another lecture which consists of active transport and passive transport, which is another part of the chapter 3, and it's already posted on Zoom, and you guys can access to it and review. Let me go ahead and